All right, ladies and gents, welcome back. So today we're going to continue our discussion on protein synthesis. So remember, we're talking about this flow of information in your cells from that genetic code in your DNA, narrowing it down to a single gene, a single set of instructions that we're then going to copy into a messenger RNA via that process of transcription, and then take that mRNA copy out into the cytoplasm to your ribosomes, and do that translation process, converting that triplet code, that three nucleotide code, into the amino acid language, the actual building of the protein itself. So for today, we're really going to just focus on this first part, that transcription piece. How do I go from taking that DNA code, that gene, and creating messenger RNA from it? And so that's what transcription is. It's writing the code and then moving it or crossing that membrane. So remember, transcription requires DNA, which means this has to happen in your nucleus. So that is the site of this particular process. We're going to make that mRNA from DNA. Once it's created, it goes through those nuclear pores, these little holes that are in the nuclear envelope or the nuclear membrane. We'll move that messenger RNA then out to the cytoplasm, to a ribosome, to actually do the translation process. So during transcription, I have to build this RNA molecule. And in a lot of cases, it's going to look similar to what we did with DNA replication, in the sense that I need to open up the DNA molecule to have access to those nucleotides, and then we're going to build a complementary strand using our base pairing rules. Now, a couple of things that are a little different here. Unlike DNA replication, where I ended up making copies of both strands of DNA, your DNA, one of those strands actually contains the information that we need. The other strand really is there just to be that other complement to ensure that I can make DNA copies during replication accurately. So when we talk about the DNA strand that's actually used for transcription, we call it the template strand. The other strand that is complementary to it in the DNA molecule isn't ultimately going to create a messenger RNA instead. So if I take a look at the diagram below, here is my template strand. Here is the actual gene coding region that creates the mRNA, and then this is the complementary DNA strand that's just there to keep the molecule together, and we're not going to access that information. So when I'm going through transcription, I need a couple of pieces. I need the DNA template strand. I also need free RNA nucleotides to build that mRNA strand, and then I need an enzyme. So the enzyme that we're going to end up using for transcription is called RNA polymerase. Much like the DNA, right? ACE is an enzyme, polymerase, I'm building a polymer, and in this case, I'm building that RNA polymer as opposed to using DNA polymerase like we did in DNA replication, where we built a DNA polymer instead. The other thing we have to remember is that your base pairing rules still apply, but remember that there is no T, there is no thymine in RNA. Instead, it's uracil. So when I'm looking at that template DNA strand, here's a T, well, it's going to base pair to A, just like we're used to seeing so far. But when I've got an A in my DNA strand, I now replace it with a uracil instead. G and C still go together just like they have. So that's really where that big difference is that we have to remember when we're copying the code that I'm taking it from A to U when I go through and create that messenger RNA. So here's the thing. When we look at your DNA molecule and we talk about those 46 chromosomes worth of information in human eukaryotic cells, human diploid cells, we're really looking at a chromosome that may contain 5, 6, 7, 8, maybe even hundreds of genes on a single chromosome, depending on which chromosome we're talking about. So in order for your RNA polymerase to know where a gene starts and where it stops, it needs some way to recognize that. So I like to think of the basic gene structure that we're looking at here in the picture similar to the basic structure of a sentence you would see on a page. There could be 10, 20, 30, 40, 50 sentences on that page, but how do you know where the sentence starts and where it stops? Where is there a complete thought? So your basic gene structure is set up like a sentence. I've got this promoter region right here. And think of it like the capital letter. That's how you know that the sentence is starting. That's where we're actually going to bind that RNA polymerase and it's actually going to start translating. On the reverse end, when we look at the end of a sentence, you have some sort of punctuation mark, a period, an exclamation point, a question mark. Your terminator region down at this end of the gene is like that punctuation. How do you know when the sentence ends? Because we use one of those punctuation marks. How does RNA polymerase know that it's got all the information that it needs to make that protein? Because it will run into that terminator sequence, and molecularly, that terminator will tell that RNA polymerase that it's done its job. And in between is the meat of the sentence. What is the sentence actually telling you? What is the information? And in the case of the gene structure, that's what we're talking about with this RNA coding region. It's the actual information that will eventually get translated into proteins. So when we look at transcription, there really are three major pieces to the process itself, or steps to it. And initiation really starts at that promoter. And here in initiation, that RNA polymerase binds to that RNA promoters region and unwinds the DNA. This is going to allow us to start attaching those RNA nucleotides and start building the copy, building that messenger RNA, so that we can take that information out of the nucleus into the cytoplasm. Elongation is exactly what it sounds like. To elongate is to grow or to build something. So once that RNA polymerase attaches to that promoter, now we're going to take these free RNA nucleotides that are floating around your nucleus. We're going to use the complementary DNA strand to build a complementary mRNA molecule, and we keep adding those nucleotides one at a time, base pairing them appropriately as we build that messenger RNA. And we continue to build that messenger RNA until we reach that terminator sequence. Once that RNA polymerase gets to the terminator, we go through this termination step. To terminate means to stop it. And so once we hit that DNA's terminator sequence, all the components of this process come out of DNA. So the DNA molecule, the RNA polymerase falls off, the messenger RNA falls off, and DNA is able to wind itself back up and be stable and ready for the next time that it needs to be copied.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
Now, here's the thing, when we're talking about messenger RNA, specifically in eukaryotic cells, there's some additional processing that ends up happening to that messenger RNA before it leaves the nucleus and actually goes through the translation process. And so, before we can leave the nucleus, a couple of things happen to that messenger RNA, one of which is this processing piece. So when we look at DNA, remember that it runs 5' to 3' or 3' to 5', depending on the direction. So your free 5' end is your free phosphate, the 3' end is your free sugar. Well, we get the same kind of directionality in RNA as well. So when we go through transcription, one of the things we do to that messenger RNA before it leaves the nucleus is add some things to either end. So on the 5' end where that free phosphate is, we add this 5' cap. It's a methylated cap, so it's a chemical group that we get added onto this end. And on the 3' end, we add this poly A tail. It's just a bunch of adenine, bunch of A's. And the reason that we add this cap and this tail is for two major reasons. One, as this messenger RNA is moving through the nucleus and then ultimately through the cytoplasm before it gets to the ribosome, there are all sorts of things that it's going to run into, whether physically or things like enzymes. And so, in order to help protect the good information in between from getting chopped into pieces before we actually get to make the product, we add this cap and this tail, so those are the pieces that can kind of get bumped up, damaged a little bit, and we're not actually hurting the good information that we need to make sure we make our product, our protein, correctly. The other thing is that once we get to that ribosome, I need to make sure that my cell orients my mRNA in the correct order. Because remember that I've got to read these 3 bases at a time. Well, if I somehow reverse this and put this message in backwards and was reading it backwards, then the entire product that I get out of that process is going to be all backwards as well, and likely not to do its job. So we need to ensure that when we get to the ribosome that we make sure we read it from 5' to 3' so that everything gets read correctly. So that's also another reason for this cap and this tail.
Before mRNA can be used by ribosomes as a template for building proteins, it must first be processed. Key steps are the addition of a methylated cap and a polyadenylated tail. Involved in processing are RNA polymerase, cleavage factors, and poly-A polymerase. Processing of mRNA begins with transcription. Soon after RNA polymerase begins transcription, a methylated cap is added to the 5' end. Transcription then continues to completion. Following completion, RNA polymerase releases the capped strand of pre-mRNA. Specific nucleotide sequences in the mRNA are bound by cleavage factors. The 3' end of the mRNA is next moved into the correct configuration for cleavage. Stabilizing factors are then added to the complex. Poly-A polymerase now binds to the mRNA and cleaves the 3' end. The complex begins to dissociate and the cleaved 3' end quickly degrades. Poly-A polymerase now synthesizes the polyadenylated tail by adding adenine residues to the cleavage site. Additional proteins then bind to the tail, increasing the rate at which it grows. When the tail reaches its full length, the poly-A polymerase is signaled to stop adding residues, and the polyadenylation process is completed. The processed mRNA is now ready to undergo splicing in preparation for translation.
The other thing that happens to that messenger RNA, in addition to adding that cap and that tail, is that we do a little bit of editing to messenger RNA in eukaryotic cells. So when we look at your DNA molecule and that gene sequence here, you've got these series of exons and introns. Those exons are the actual expressed pieces, the actual good information that your cells will use in order to produce the protein that it wants. But in the actual gene sequence itself, you've got these exons that are actually interrupted by these intron sequences. So exon is expressed, intron is interruption. So when we go through and produce this messenger RNA, before it's a final complete process, we have to do some editing. So that editing process there is really as a way to take out these introns and then splice these exons back together. So sort of think of it like this. You had to write an essay for your English paper. You waited to the last second to do it, and so you ended up fluffing it up with information that you knew from other courses because you never really got to do the research for that particular essay. So you hand that to your English teacher goes, all right, there's all sorts of fluff in this paper. I know you didn't necessarily do the research. I want you to pull out that extra information, that fluff, and actually just give me the information that you have. I want to see the real sort of raw research you did. So those introns are kind of that extra fluff you pulled from everywhere else. The exons, if we were to narrow it down and just include the information you actually researched, that's what you've got. So kind of think of it maybe like that. So perhaps you're thinking, well, why would my cells bother to do this? And the real reason here is because there's this process that goes on in eukaryotic cells called alternative RNA splicing. So go back to that idea that we talked about all the way on the first slide of this PowerPoint. And that was this idea that there is a single gene that codes for a single protein. And I mentioned to you that there's some caveats to that. Well, here is one of those caveats. So when we look at prokaryotic cells, single-celled organisms, no nucleus, that one gene, one polypeptide holds up every single time. But when we talk about eukaryotic cells, think about the sheer mass of information that needs to be able to fit into your nucleus in order to produce all of the proteins that your body needs in order to give you its structure and do its job. That's insane amounts of information. So if we needed a single gene sequence for every single protein that your body produces, the ability for that information to actually get packaged into your nucleus, into your cell, would be virtually impossible. So evolution over time has produced this process called alternative RNA splicing, where I can take a single gene sequence, and depending on the way that I splice up those exons and put them back together, I can make slightly different proteins that do slightly different jobs in your cells. So I can take a single gene, and now rather than one gene to one polypeptide, I may get one gene to three polypeptides. Those three polypeptides are similar in their structure, similar in their function, but they're not necessarily identical. So if I look at this image here, I've got three exons, right? Well, perhaps for protein A, I use exon 1 and exon 2. For protein B, I use exon 2 and exon 3. For protein C, I use only exon 1, exon 3. So there's a bunch of different ways that I can splice those exons together in different arrangements to give me slightly different proteins. And that's really this idea of RNA splicing and being able to sort of maximize the amount of information that I need to fit in my nucleus in order to still produce all of the varying proteins that my cell needs.
The following animation will describe the process of RNA splicing. an important step in creating the mRNA that is involved in protein synthesis via the process of translation. Key factors in this process include RNA, possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.